Hello everyone, how are you all? कैसे हैं आप सभी लोग? I hope you all must be having a great time. This is your faculty Ankit and I hope you must be connected throughout this unit. This unit 1 is going off paper number 2, that is cell biology and genetics. This is going to be lecture number 4. Over here we are going to cover these topics now: chromosomal aberrations, polyploidy, euploidy and aneuploidy. This is your syllabus as it is, and moving ahead you are going to find that cell structure function, and in the last class we have covered this cell cycle also. Synthesis, structure, function of genetic material, last class it happened. Chromosome structure also we have covered. Now for today's we are going to cover this chromosomal aberrations, polyploidy, euploidy and aneuploidy over here in this particular lecture. This is in one go we are going to cover. In the next class you have to understand the laws of heredity. This is how the procedure should be. Right, I hope you must be liking it, you must be enjoying the subject. A lot of non-agri students are also with me. I hope you are not fearing this subject, guys. Okay. Now let us do a quick recap of what we have seen till now. So our first class was about the cell organelles, their structure and their function. So over here cell theory we have read, and different organelles we have read. Then over here in the next class we have understood about the synthesis, structure and function of genetic material. Very important, very elaborate lectures I have taken for this. Okay. So what you will be finding over here, you have understood about the DNA theory, DNA evolution, DNA replication method, DNA to protein synthesis you have understood, right? All these you have understood in this particular lecture. The last lecture you have understood about this chromosome structure, types of chromosome, what kind of chromosome we have, under this how this chromosome formed, right? Formation of this chromosome, nucleosome theory I have told you. Then also we have understood over here the cell cycles, mitosis and meiosis. I hope you have understood it. I hope you will be revising it. Pause the video and just revise the cell cycle, guys, because right now there will be use. There was lactotine, zygotine, pachytine, diplotin and dikinesis. ये थोड़ा सा, this particular part is a bit complicated to remember. Otherwise else is nothing but PMAT, है ना? I hope you must be understanding, guys. Now we will be moving ahead and we will be seeing this chromosomal aberrations. The changes, aberrations means the abnormalities, the changes which are happening in chromosomes. So chromosome aberrations, over here you see the abnormalities which are basically happening. Chromosomal aberration, that means there is certain changes which are taking place in this chromosome, and but obvious there must be some changes which are taking place in this chromosome, because of which the progenies are different, because of which no two species or no two progenies are similar. Parent to daughter also there will be dissimilarity. So these are happening just because of these chromosomal aberrations. Now these chromosomal aberrations arises of two things. One, based upon on the position of chromosome, position, right? And position means, that means the chromosomal structure is getting change. Structural changes you are going to find. Structure you must be knowing of the chromosome, that is changing, that there is some change in that. In structural changes you will be finding either the genes, there is a change in the gene number, which are present in the chromosome, in the DNA, or you will be finding change in the gene sequence. This table is very important. Change in the gene sequence, that I'll be explaining you. Change in gene number, change in gene sequence. Understand very carefully. Right, so what you can find in change in gene number means either the gene can be increased, that means if plus I am showing that the gene number has increased somehow in a chromosome, or a gene number can be decreased, somehow the gene number has been reduced. So for positive I'll be calling addition. For the subtraction sign for the gene is reducing, I'll be calling it deletion. And this addition is nothing but also you can call it as a duplication. Duplication of gene happens. And deletion means that gene has been subtracted over here. Very, very important, guys. Very, very important. Okay? This is one chromosomal aberration. Deletion. This is a one chromosomal aberration which you are going to find. Now, change in gene sequence, what you are going to find, either the gene has got inverted, so that will be inversion. I'll be telling you what is inversion and all, we will be discussing. Or the gene has got translocated to some other places. So translocation. This is your another chromosomal aberration. Okay, so what you are understanding that, let me just make it same. Okay. What you are understanding over here is that chromosomal aberration happens by one way, that is the structure of chromosome is changing. And if you can remember the structure, I hope the structure you are remembering, guys. The structure somehow where you have found like this, and this is also there is secondary constructions you will be seeing. This is centromere you will be finding. This is nothing but your DNA, chromonema, and over here DNA is coiled up. Over here genes are present, right? Like this you will be finding genes are present, है ना? So if there is a change in gene number, the gene has been got added, duplicated, that is duplication, deleted, deletion. The gene has got transferred from one location to another, translocation. The gene had got inverted, inversion. Alright. So this is one kind of chromosomal aberration that is based upon the structure. Basis is structure. Now other one is your numerical changes. Numerical changes or aberrations also you can call. Numerical. What is numerical aberrations? You will be understanding, guys. ठीक है, इसको duplication ही रखना है यहाँ पर, addition छोड़ दो ना, addition क्योंकि यहाँ पर पढ़ोगे आप तो इसे duplication ही नाम दो, confuse हो सकते हो, duplication. In numerical aberrations, what is going to happen? Numerical aberration is known as, first of all, heteroploidy. Heteroploidy. In this, what happens? There are two types of conditions. One condition is of aneuploidy. Second condition is of euploidy. In aneuploidy, what happens? That there is addition or subtraction of a particular chromosome. Now let me explain you. Okay? Under aneuploidy, just see the structure first of all, everything which I have to explain you over here. In aneuploidy, what you will be finding that there is a one chromosome which is being added. So addition of chromosome might be happening over here. Addition of chromosome or a deletion of chromosome will be happening over here. A particular chromosome you are going to see. So there will be multiple example over here you will be finding, that is monosomy, double monosomy, trisomy, double trisomy. Like this you are going to see over here and I will be explaining. Euploidy is a condition where the whole set of genome is being added. That I will be telling you again. Over here the condition emerge as of haploidy and polyploidy. This also we are going to study in this particular lecture, and the topics which are containing are heading as polyploidy, euploidy, and this particular aneuploidy, right? What the topic name we have mentioned over here? Polyploidy, euploidy, and aneuploidy. ठीक है, तो ये सारी चीजें हम इसमें समझने वाले हैं structures के through. Everything in detail we are going to understand. ठीक है, one by one let us understand. Let us see the first structural type of numeric chromosomal aberration. In this we will be understanding the deletion first, guys. ठीक है, let us see the deletion first over here. Deletion. This is structural changes which is happening in a chromosome. This is your chromosome, guys, over here. Okay, and in the chromosome we have genes like this: 1, 2, 3, 4, 5, 6. Suppose these are my genes. Like this also we will be having the genes: 7, 8, 9, 10, 11, 12, 13. Like this, any number I am putting. Deletion is a process in which what happens that somehow the part of a particular chromosome get cuts off. For example, if there is one break happens, So this 5 and 6 get cut off over here. 5 and 16 got cut off from here. Right, this is one type of deletion which might be happening. Or sometimes what can happen, that in between that 3 4 get cuts off. So that means for 3 4 to get cuts off, you need 2 breaks like this, and then this 3 4 will be cutting off from here, 3 4. So the deletion which has happened of a terminal part is known as terminal deletion. The deletion which has happened of the internal intermediate part, we call it as intercalary deletion. Right, like this you will be finding in deletion. Now, why deletion happens? One of natural causes, because of UV lights. That's why we say that UV hampers with the DNA, right? It mutates. Mutation means change. Again, in separate lecture, we have to see that how it happens. But these are things are happening also natural and can be induced also by UV rays, X-rays, thermal induction we can put, electrical induction we can put. All this will be understanding in mutation, but this type of thing is happening in the deletion case. Try to understand. Sometimes what happens, the deletion happens is of homologous deletion also you will be understanding. Suppose this is your particular chromosome like this. Homologous deletion means 2 parts are getting cut off at the same point of time. And heterologous means 1 part of here and 1 part of another chromosome. Simple. So these are the type of deletion. I don't think they will be asking you in detail about this. They have never asked this also. But this way can be asked. Also you understand that in deletion, if I draw it on the chart, like this, 1 2 3 4 4 5, then there is, suppose this is the centromere, 6 7 8 9. Now let me show you that, let me mark it again in this manner only, that this is your 1 2 3 4 5 6 7 8 9. This part I have shown you of the chromosome like this. Now I am telling you that 8 and 9 got missed out over here, or 7 or 8, 6 or 7 got missed out over here. Okay, this has been cut over here. So what my chromosome will look like now? 1 1 2 3 4 5 6 7 I have got missed out, 8 and 9. This has been cut. I am just telling you for it for the explanation purpose, nothing else, for your understanding. This will not be asked, neither you have to show it also. Now, if they necessarily ask you that what is the procedure of duplication, deletion, and what happens in deletion. Now, what will be happening, you will be finding over here that when this type of particular thing is there, because always we have in pair, these chromosomes are always in pair, and in one chromosome you are finding that this intercalary deletion has happened, this 6 7 is not there. So what is going to happen, that these pairing will be happening, and I have taught you cell cycle now. In meiosis, you will be understanding synaptal complex forms. Both of the chromosome joins like this. So when they are joining, they will be pairing with the same gene. 1 1 2 2. Pairing of the same gene happens. 4 4 5 5. But you will find 6 and 7 is not there. Directly 8 and 9 is there. So what happens is this particular 6 and 7 will be forming the loop like this. So 6 will go over here. 7 will go over here. And it will be forming like 8 9 now will be coming like this, so that they can pair with the 8 and 9 part. And that's why in deletion, most of the times scientists have observed that a deletion loop is being formed. When a loop is being formed, it is a one of type of confirmation that over here some deletion has happened. So this is the nature's way of doing things. Okay, nature will form a loop over here, and let me show you too. Dekho. Before deletion, one particular part, this is the deleted area. After deletion, this happened. This is a normal chromosome. This is a deleted chromosome. What is going to happen? C and D will be forming the loop. E and F will be combining. A and B will be combining. Are you understanding? Like this, a deletion loop gets formed. I hope you are understanding. So I have told you about the deletion. There can be terminal deletion. There can be intercalary deletions, right? 2 breaks are needed. You can explain it like this, like the way they have explained over here, or you can draw the diagram also. No, not very difficult to draw it. Packetine stage of meiosis, I hope you are understanding what is happening at packetine. Okay, packetine, you will be understanding at this point of time it can be detected because loop formation happens. Okay, it can be induced by radiation or mutagenic chemicals. What are mutagenic chemicals? I'll be telling you in the mutagen class. Consequences are lethal. Only essential gene loss, lethal mutations can happen. Right? We have a famous example over here, Drosophila. Drosophila deletion found on X chromosome, which involve W white eye, facet eye, and vermilion eye color. Right, so you will be finding barred eye type of feature in this. What happens because of deletion? Chromosomal mutations, variability, and speciation you will be finding. Second is your duplication. Now what happens in duplication also you understand. Suppose this is your particular chromosome. One, two, three, four, five, six, seven, eight. What is going to happen? That sometimes the deleted part which was chopped off of another gene, suppose this is containing your 3-4 chromosome of another particular chromosome. So this 3-4 will be coming and will be getting attached over here like 3-4. If it is getting attached in the same manner, we will be calling it something with some name. If it is, we will be calling it as tandem duplication. If it is attaching in a reverse order like 4-3, then reverse tandem duplication. If it is on the same line, same arm, then we will be calling it something. If it is on the different arm, then we will be calling it differently, right? So like this, you are going to find a lot of these types of duplication, that is repeats. Duplication, nothing, segment of a chromosome present more than once. So if the sequence is like this, just wait, really sorry. I hope this is visible, just wait. Now you see, they go tandem. If the sequence is right after the original in same orientation, A, B, then B, C, 1B has been copied. Reverse tandem means extra segmented is inserted but in the reverse order, A, B, B, C. Or rather than you highlight it like this, that the whole AB got copied, है ना? AB, AB. Reverse tandem you want to say AB, BA, C then, right? Like this you can show. Displaced means it is inserted from original in same orientation. Reverse displaced means extra segment is inserted away from the original in reverse orientation. For example, this is nothing. Displaced is 1, 2, 3, 4 and 5 over here. Suppose 3, 4 you have to duplicate it. So I have told you either in tandem or reverse tandem. What if 3, 4 gets installed over here? 6, 7, 8 and then 3, 4 again comes over here. This is displaced. So see this, displaced and reverse displaced means 3, 4 not 4, 3 like this, right? So like this you will be finding these are the type of duplication, simple to understand this. Okay. Now because of duplication what you will be understanding, there will be one category which will be getting formed and this is known as, I have told you that there are types of chromosome, and types of chromosome I have explained you that there is one type known as dicentric chromosome, tricentric chromosome. What do you understand by dicentric and tricentric? Now let me tell you this. Okay, now because of duplication, suppose this is your 1, 2, 3, 4, 5, 6, 7, 8. Normal chromosome is this. Now there is one duplicated chromosome 1, 2, 3, 4, 5, 6, 3, 4, 7, 8. So duplicated is displaced type of chromosome you are seeing over here, displaced. Now what is going to happen that these will be combining with each other. Now rather than this 3, 4 combine with this particular 3, 4, this 3, 4 sometimes get combined with this particular part. With this particular part it will be combined. And because of this, when it will be combining with this particular part, sometimes a chromosome get deleted. And sometimes the situation will be like this, that the chromosome will be added also. So double chromosomes you are going to see. Okay, for example, suppose this 3, 4 is not here, but 3, 4 it get added over here, right? Suppose 3, 4 get added over here. So what will be happening? This 3, 4 will be joining like this, है ना? इसमें आपको डबल नहीं दिखेगा, डबल में दिखाता हूँ. देखो यहाँ समझने की कोशिश करता हूँ. Again I am taking 1, 2, 3, 4, 5. This is your centromere. 6, 7, 8 और यहाँ पर suppose 3, 4 दोबारा आ जाता है, ठीक है? 3, 4. This is 8 over here. Now I have this particular part as my original one. 1, 2, 3, 4, 5, 6, 7, 8. Now what is going to happen is that this line which is going like this, it will not be matching up with this, rather it will be matching up with this 3 4. So what is going to happen over here? This line I am drawing 1 2 3 4 5, one centromere. Then this is not joined over here, this will be joining with this particular part, right? So what is going to happen? So this, because of this joining, it will be appearing like this, that 3 over here, 3 4 will be coming, 3 4 will be coming like this, है ना? ये ऐसे join होगा इसके साथ, and then it will be joining like this. Then you will be finding that one more time this particular chromosome will appear because of this connection which is happening. So double, tri or dicentric you will be finding. ठीक है, I hope you have understood. But इसमें एक प्रॉब्लम अभी ये है कि आपको शायद ये ढंग से समझ में ना आया हो, क्योंकि मैंने क्लियरली आपको ड्रॉ नहीं किया, तो एक बार मैं क्लियरली आपको थोड़ा बता दूं। दो इसकी रिक्वायरमेंट नहीं है, ना ही कोई पूछेगा, लेकिन एक बार क्लियरली बताना मेरा काम है, तो मैं आपको बताता हूँ क्लियरली। 5 दोबारा हम इसको समझेंगे। 6 7, सपोज दिस 3 4 इज ओवर हियर, आई हैव टोल्ड यू 8 9 तक ले लो, ठीक है। देन देयर इज वन नॉर्मल क्रोमोसोम यू कैन ड्रॉ ओवर हियर। अब इसको ऐसे ड्रॉ मत करो, जो मैं ड्रॉ कर रहा था यही प्रॉब्लम हो रही थी। सपोज ये नॉर्मल क्रोमोसोम है ना, 1 2 3 4 5, ये सेंट्रोमेयर 6 7 8 9, ड्यूप्लीकेशन इसमें हुआ तो नॉर्मल कौन सा, नीचे वाला। ये ऐसे अटैच नहीं होगा, ये अटैच कैसे होगा, तभी आपको कन्फ्यूजन हो सकती है, क्योंकि मेरे को भी कन्फ्यूजन हो गई थी। ये अटैच ऐसे होगा, ठीक है, ये लाइन यहाँ जाएगी ऐसे करके, ठीक है। अब ये क्या होगा, 1 2 3 4, वाई 3 4 इसके नज़दीक क्यों आ गया, क्योंकि ये मैच करेंगे यहाँ पर, ठीक है, मैच करेंगे। बाकी क्या आपका 5 नॉर्मल है, तो 6 7 8 9, तो इसका सेंट्रोमेयर कहाँ है, यहाँ पर है। अब आई होप आपको क्लियरली समझ में आ जाए। इसका दूसरे वाले का नॉर्मल का सेंट्रोमेयर यहाँ है, डुप्लीकेटेड वाले का यहाँ है। तो अब आप ये देखो कि मेरा अपीयर कैसे कर रहा है, ये वाली लाइन देखना। तो मेरी एक लाइन ऐसे नजर आएगी, ये जा रहा है, ये जा रहा है, ये जा रहा है, 3 4 कंबाइन हो गया, तो ये एक मेरी पर्टिकुलर क्रोमोसोम नजर आएगा मेरे को, ये चीज। आई होप यूर अंडरस्टैंडिंग। सो अगर मैं इसको ऐसे ड्रॉ करना चाहूँ, दैट दिस इज गोइंग लाइक दिस, एक सेंट्रोमेयर, दिस इज गोइंग लाइक दिस, एंड दिस इज गोइंग लाइक दिस, वन मोर सेंट्रोमेयर। कैसे, 1 2, सिर्फ रेड को जो मैंने मार्क कर रखा वो देखना ऊपर, 4 5, एक सेंट्रोमेयर आ गया, फिर 6 7, ओवर हेयर क्रॉसिंग ओवर हैज हैपेंड, इसे क्रॉसिंग ओवर बोलते हैं। और मैं बाद में आपको जब हम क्रॉसिंग ओवर का देखें तो डायरेक्टली एप्लीकेशन बताऊंगा ये हो कह रहा है, दीज आर हैपनिंग बिकॉज ऑफ दीज पर्टिकुलर क्रोमोसोनल एबरेशंस। 3 4 आ गया, देन आफ्टर दैट 5, and after that one more chromosome is there, or centromere is there, 6 7 8 9. So this is the dicentric situation which you are finding over here. दूसरा टाइप का कैसे बनेगा, इसमें दूसरा टाइप का आप यहाँ फोकस करना ब्लू पार्ट को, 1 2 3 4 लाइक दिस, 8 9 लाइक दिस। तो ये कौन सा बना, इसमें क्रोमोसोम आ रहा है क्या, कोई भी सॉरी सेंट्रोमेयर आ रहा है कोई भी, नहीं, तो ये एसेंट्रिक टाइप ऑफ क्रोमोसोम बन जाएगा। I hope you are understanding. I'm trying my best so that you can understand this much. ठीक है, so because of this duplication you find this bar I in Drosophila. इसको आप as it is याद रख सकते हो। It is happening because of the duplication of region 16A on the X chromosome. This is important for your example. ठीक है, next you understand the inversion. Very important, very interesting. देखो, after understanding these type of aberrations you will again go to my types of chromosome. In types of chromosome I was saying you that this you will be understanding later on in the reasons why these types are created, dicentric, acentric, monocentric. Now you must be understanding, because in that particular reason they have written inversion, paracentric inversion, all this they have written. So now you will be understanding these inversion things. So you understand over here you are having this particular chromosome inversion. This is 1 2 3 4 5 6 7 8, 1 2 3 4 5 6 7 8. Now in inversion what happens, basically what you are going to find, that this, suppose this 3 4 over here will get inverted. Inversion means there will be change in the position of the gene. So position of the gene, how it will be changing, 3 and 4 will be getting converted into 4 and 3. So this will convert into 4 and 3 like this, right? When it is happening on one side of a arm, then it is a paracentric. Now sometimes this inversion can happen including this centromere also. For example, let me show you. Suppose this 4 and 5 can also get inverted. हो सकता है, नहीं हो सकता? हो सकता है बिल्कुल. So this 4 and 5 will look like 4 over here and 5 over here, and in between that there will be centromere involved. So when centromere is involved, we call it pericentric. So there are two types, pericentric and paracentric. Very easy to understand. Pericentric and paracentric inversion. Because of this inversion, you will be finding that there are certain things happening which is crossing over. Because of crossing over, you will be finding there is modification in chromosome happening. That's why we are saying that there is a chromosome aberrations which are happening. And the most important thing which you will be finding over here is not the loop formation. Because देखो, one thing you are understanding that genes are having a feature of matching with each other. Two chromosomes will be matching with each other. So because of this, what is going to happen, and this happens in synapses in the meiosis. Because of this, what will be happening over here, that suppose now I have two genes over here, guys. Let me highlight again. Similar gene I am showing you. Okay, in reverse condition I am showing you, right? 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 4, 3. This got changed. So let me highlight it by red color. 4, 3. Okay, this is normal. 5, 6, 7, 8. So what is going to happen that this 3, 4, 3, 4, how this will be matching now? Now if you will be putting it on the line 1, 2, 3, 4. This is your centromere. 5, 6, 7, 8. There is one more thing. 1, 2, 3, 4. But there has been the duplication of 3, 4 has happened ना, तो 4, 3 ये बन गया ना, तो इसको रेड से हाईलाइट कर लो. ये तो 2 है, इसको 4, 3 लिख लो. ठीक है, ये शो ऐसे ही आप एग्जाम में कर सको तो करना. You will be getting extra mark 8. What will be happening over here, that now this everything has to match up. Everything will be matching up except 3 and 4. So how they will be matching up over here? So they will not be forming a loop. There will be forming an inverse loop over here. How? That this 3, 4 will be now getting looped in this way. In this way. Right? So this 4 will be going over here and this 3 will be going over here. देखो, ये समझने की कोशिश करो. ये इस वे में लूप बना लेंगे. अब ये होने की वजह से क्या होगा, 4 इस साइड आ जाएगा, 3 इस साइड आ जाएगा. And then they will be matching over here. This match will be happening. Right? So one time they will be looking like this. Second time they will be looking like this. ऐसे करके chromosome बनेगा. Understanding easy to understand, guys. ठीक है. So over here you देखो, या तो mechanism ऐसे समझा दो, but if I'll tell you to make them understand by the diagram, schematic diagram, you can make it. Definition is nothing. You can see segment get flipped by 180 degree. This is understanding is important. Paracentric, centromere involved? No. Pericentric involved? Yes. Like this you can show. So this loop, inverse loop, which I was telling you how it is getting formed. This is normal one. This is the loop which has got formed. इसमें क्या करा है इन्होंने, BCD की पोजीशन चेंज हो जाती है DCB में. So they have formed the loop like this, DCB. Why loop? Because BCD has got transferred into like this. That's why there will be a inverse loop which is getting formed. I hope you are understanding, guys. This is an example of pericentric and paracentric. Okay. Now see this translocation, guys. Translocation. Translocation is the shifting of genes from a homologous to non-homologous. Okay. This is a shifting of genes to non-homologous chromosomes. ठीक है, suppose these are two chromosomes of different these are two non-homologous chromosomes you are finding. Non-homologous, I hope you are understanding. This is homologous over here, and other chromosome which is present, because in any particular type, for example in human you are having 46 chromosomes, 23 pairs. In pairing you will be finding the pair will be of homologous chromosome, other pair will be of other chromosome, so that two will be called as a non-homologous. So if gene is getting translocated from one to another part of the chromosome, that is called as a translocation. For example, let me tell you, 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6. Now this will be same in the homologous category 1 2 3 4 5 6 same रहेगा, right? But in non homologous the number of gene will be changing obviously. So 1 2 3 4 5 6 यहाँ पर 7 8 9 10 11 12 you are going to find different gene. Now this will be same 7 8 9 because this is homologous for them, this is homologous, but these two are non homologous. So what will be happening over here is that suppose this 6 5 4 get transferred to this particular area and 10 11 12 will be coming to this area. So this will become 10 11 12 and this will become 4 5 6. So what change I have shown you, again I am telling you 1 2 3 4 5 6 7 8 1 2 3 4 5 6 7 8. Suppose this is a non-homologous 9 10 11 12 9 10 11 12 13 14 15 16. Now suppose this got translocated 5 6 7 and 14 15 14 13 get translocated. That means now the new after translocation this 13 14 15 comes over here and 5 6 7 comes over here. This is nothing but your translocation. Now there are different types also you will be finding under that, but not needed. In the PPT I have given you that too. But because of this thing, what you will be finding that there is a unique structure which is getting formed because they will be having to match over here. Because this is the matching will be happening of the homologous pairing. So there is one particular chromosome like this, homologous will be there, and these two have to match with each other. So how they will be matching? So they will be forming a different shape over here, guys, and a very unique shape you will be finding over here. Suppose, let me draw it like this. I'll try to explain. Let me see if you will be able to understand or not, or if I'll be able to explain you well or not. 1 2 3 4. This is one centromere. 5 6 7 8. 2 3 4 5 6 7 8. 1 2 3 4. This is going to be same because these are homologous. 4 5 6 7 8. ठीक है, दिस इज वन होमोलोगस क्रोमोसोम. ये एक है ना, तो मैंने ये दोनों बना के दिखा दिया आपको. ठीक है, अब दूसरा एक बार क्रोमोसोम में बनाने की कोशिश करता हूँ. ये आपका नॉन होमोलोगस कैटेगरी के आ जाएगा, लेकिन रहेंगे तो ये भी सेम ही. तो ये दोनों आपस में होमोलोगस हैं, बट when there will be crossing over happening over here, then they will are non homologous over. ठीक है, this is your 9 10 11 12 13 14 15 16. ये भी सेम रहेगा. Now till now translocation happened or not? No. This is one chromosome, homologous chromosome. This is different chromosome with different genes on them. Okay, now what happened? Translocation happened. That means suppose I do the translocation of these particular pair over here. So what will be happening? This will be getting converted into 13, 14, 15, and this will be getting converted into 5, 6, 7. Now how these things will be matching up now? This is a very unique to know, because I told you that genes has a habit of matching, and that too in synapsis stage. How this will be happening? To match with the you will be finding in the deletion you are getting the loop, in inversion you were getting the inverse loop, in the duplication you were getting the formation of dicentric tricentric. Over here you will be getting the formation of the plus shape over here, right? Now it is getting formed. Try to understand. I'll try to explain, guys. देखो ये कैसे बनेगा. ये दोनों तो मैच करेंगे ही करेंगे, तो आप इसको इस तरीके से बनाओ. 1 2 3 4. This one over here 1 2 3 4, because these will be matching. The difference is where this particular over here I am highlighting, and this will move like this 5, and this is centromere 5 6 7 8, right? And this will move this side, and what is there? 13 14 15, the translocated one, and you are getting 8 over here. Now you try to understand. Now what is going to happen? Now you come with these two particular part, this particular chromosome. How they will be getting represented over here? Because 9, 10, 11, 12 will be matching. So draw like this. 9, 10, 11, 12. 9, 11, 12. They will be matching and they will be moving like this. देखो यहाँ पर 4 सेंट्रोमेयर present हैं, तो इसकी वजह से भी आप देखोगे यहाँ पर quadrupled centromere you will be finding. And like this, like this. Now you tell me what should come. You automatically will be telling me. Over here, 9, 10, 11, 12, 5, 6, 7, 8. 5, the inverted one, 7, and 16 happened over here. But this will be matching over here ना. ये 16 है, but ये match कर गया यहाँ पर. ठीक है. अब यहाँ पर क्या है आपका 13, 14, 15. So यहाँ पर 13, 14, 15 आएगा. 13, 14, 15. So this will be matching in this way. So a structure type of a quadruple type of thing has been formed over here. So I hope you have understood. यहाँ पर मेरे पास इसकी इमेज नहीं है, but if you will be searching it on net, you will be getting it, है ना. इसको एक बार गूगल इमेजेस में देखना, आपको जरूर मिल जाएगा. बट आप इसी तरीके से शो कर सकते हैं इसको. So I have told you the deletion part, duplication part, inversion part, translocation part, and translocation two non-homologous chromosomes are shown. See, whereas these are homologous. I hope you have understood the difference between this homologous. This is one pairing which is being done. This is your homologous chromosome. Okay, whereas another one over here, non-homologous. Different genes are present on this particular part. The genes are same on the homologous. The genes are different on the non-homologous. Okay. So in our table of structural changes, we have seen change in gene number, that is duplication, deletion, change in gene sequence, inversion, and translocation. I hope you have understood this. Now we are going to understand the numerical changes, aberration. This is also very unique to understand, guys. Heteroploidy. That means now the number of chromosomes are changing over here. Let us take the example of P. Okay, I am taking the example of P so that I can explain you. 2n is equal to 14. That means I have 7 pairs. That means over here, if I draw like this, 1, 2, 3, 4, 5, 6, and one more, 7. Now sometimes what happens in nature, that sometimes you will be finding that over here one particular chromosome will be getting added like this, है So what happens if this particular one chromosome is added? You can say that 2n is equal to 14 में क्या हुआ है, +1 हुआ है, right? And this is nothing but your trisomy, because over here triple chromosome have formed. Sometimes one more chromosome is added, but on the second chromosome number 1, 2, 3, 4, 5, 6, 7, second pair mein. So one more you will be writing and you will be calling it as double trisomy. Sometimes what happens that one full chromosome get added, not like this, but a full chromosome pair get added. That means now you will not be terming it as this, but you will be terming it as +2. Then you will be calling it as a tetrasomy. Okay, this is one type of condition. Double trisomy, trisomy, tetrasomy. Now in one type of condition, what you will be finding, देखो condition अभी समझा रहा हूँ मैं आपको. One type of condition, what you will be finding, that this particular part gets deleted. Okay, suppose this particular parts get deleted over here, this get deleted. So this is nothing but 2n minus 1 happened, है ना? In another also this can get subtracted to -1, -1. So this -1 is called monosomy because single is remaining, and one more happened, then it is called double monosomy. But instead of this particular part which is getting subtracted, what if a whole chromosome, whole pair gets deleted? That means now 2n minus 2 has happened. 2n minus 2 has happened. So this is called as nullisomy. That a whole pair got deleted over here. So these are the type I am telling you in aneuploidy. In heteroploidy, there is one condition like aneuploidy where the number of chromosomes are being added or subtracted. यहाँ पर क्या मैं क्रोमोसोम को सब्ट्रैक्ट कर रहा हूँ और ऐड कर रहा हूँ. 2n is equal to 14. 14 is the number of chromosome. 1 2 3 4 5 6 7 8 9 10 11 12 13 14. 7 pairs are there. ठीक है. Now there is one condition. This is one condition where chromosome number is getting added or subtracted. Now one condition is also there, but you will be finding in that condition that what if my whole genome set is getting added, duplicated. So what if 7 pairs is also coming? 7 pair got duplicated like this. Again, generally this happens in the interphase stage. Duplication happens. What if my whole chromosome gets duplicated over here? That means now 2n is 14 plus 2n is equal to 14 is getting added, or this particular pair is getting added. That means it has become 3n now. 3n ban gaye because 14 plus 14 you are getting 28 chromosome now. Like this 4n can also be happen, 5n can also be happen if it gets duplicated, and this generally happens in the plant species. We get tetraploidy and more than, and sometimes this can, or sometimes this particular chromosome numbers gets half number हो जाता है. Half number कब होता है? And you know this happens in meiosis. Only 7 will remain, and this happens in meiosis. N is equal to 7, and this stage is called haploid. What if double get added? It gets 3n, 4n, 5n. But in the aneuploidy, now let me tell you the terminology for it, है ना? Now you understand. In aneuploidy, what was happening that your chromosome was getting increased or decreased, plus minus 1. In euploidy, you will be finding the whole chromosome set is getting added or subtracted. Understanding this? Now let me show you. देखो, numerical aberrations you are understanding under this euploidy, aneuploidy condition. Aneuploidy refers to numerical chromosome aberration involving addition and deletion of one or more individual chromosome will be your terminology, not entire set. Because entire set अगर आ जाएगा तो then it will be coming under the euploidy. So what if one chromosome, now you remember this structure which I was showing you. थोड़ा पीछे जाओ, इसको delete कर दो, 2n minus 1. थोड़ा पीछे जाओ, 2 को delete कर दो another chromosome set के, तो 2n minus 2. सॉरी, दोनों का अगर एक ही पेयर माइनस हो गया तो 2n minus 2 that is nullisomy. So monosomy 2n minus 1 you will be finding over here. Example wheat is very famous. Under this variation of double monosomy, triple monosomy you will be finding of 2 non-homologous chromosomes. This is very important terminology, guys, which I am saying that this particular 2 non-homologous got deleted like this. What if a homologous pair get deleted completely? That is nullisomy. Okay, variant double nullisomy, double monosomy also you can get over here. What if addition of chromosome is happening plus 1? That is trisomy. Variation you will be finding double trisomy or triple trisomy. Example datura you can take over here, right? The example you will be finding that 2n plus 1 25. In human also this creates certain problems. You will be finding the certain syndromes because of this, right? Tetrasomy is 2n plus 2 where the whole homologous pair gets added. Okay, so this is an example over here: monosomy, nullosomy, trisomy, tetrasomy, double monosomy, double trisomy. But you make it in a chart form the way I have told you. Okay, so this is simple. Now its application in genetics and breeding, you will be finding gene mapping, chromosome substitution, study of doses effect, model of chromosomal orders. This you will be better understanding when you will be doing plant breeding too. Okay, and in enrichment also we'll be discussing. That's why it is very important for you to go through the units once, then only a holistic knowledge will be developed. Now in your knowledge base, certain things will be making sense in the 4th lecture. From 1st lecture to 4th lecture, things must be interconnecting. That why in the types of chromosome you are seeing this chromosomal aberrations happening because of this. Right now also you don't have answer that why this is happening, that you will be getting in mutation. This is nothing but mutation which is happening, है ना? Mutation, how it happens, what are the different types, this you will be getting under certain other head where we will be discussing that. Now there is one thing that is euploidy. In euploidy, I have told you there might be monoploid, diploid, or polyploidy. That means diploid is nothing but V organism 2x, polyploidy is 3x, 4x, 5x, monoploidy is x, and sometimes haploidy is also x, right? The famous example for monoploid is only and only maize you will be finding in nature. 2n is equal to 20, x is equal to 10. So monoploid is 10. Whereas haploids are having individuals where gametic chromosome number of a species is half. Okay? So polyploidy is nothing but the presence of more than 2 complete sets. For example, this is 2n is equal to 14, one more set has come. 3n is equal to one more set has come, that is 14 over here, right? Or 7 over here. देखो यार, इसको simple समझो. 2n is equal to 14 if for the p I am saying. ठीक है? That means pair is 7. 7 pair is that. That means 7 pair are getting added over here again. So that means 14 chromosome have added. 3n is equal to 14. 28 will be happening over here, right? Like this, you will be finding that if you keep on increasing, then this is triploid, tetraploid, hexaploid, octoploid. In hexaploid, tetraploid, you will be seeing hexaploid example, bread wheat, banana, famous example of triploid. Now how you will be giving this polyploidy, there can be artificial ploidy can be happening, physical agents, no need to remember right now, we will be doing it in the enrichment also. When you will be understanding, we will be having the knowledge of mutation, plant breeding also, then it will be more useful. Chemical agents again I'll be telling you fixed chemical agents in mutation which you have to remember. Okay. Then over here you will be finding importance in plant breeding, production of seedless fruit, increase in size and vigor. एक बार अभी read कर लो, again you will be seeing it in when we will be completing plant breeding. Now there are different types of polyploidy. This is very important right now for you to understand. There is one autopolyploidy, allopolyploidy, these are important. Then there is segmental alloploidy too. Autoploidy means these are multiple organisms where multiple chromosomes are identical. Okay. For example, you will be finding over here that A, one more set is added A, one more set is added A. That means there is nothing new chromosome is which is being added. Autopolyploids. Self, same genome repeated. A is the genome, A is getting repeated every time, right? Whereas allopolyploidy is a polyploidy organism that contains chromosome set from 2 or more different species. This is genetically distinct and non-homologous. Suppose A and then BB is getting added. Like this you will be finding. Now there are n number of example of alloploidy which you have to remember. Cotton, this you have to remember. Tobacco, okay, or wheat. I will suggest you to remember wheat and tobacco over here. General examples, okay, and they might be asking you that too. So there are 2 species, and Sylvestris and Tomentos. 2n is equal to 14, 2n is equal to 24, 2n is equal to 24. This form 2n is equal to 48 over here, which is N tobacum. Right, experimental hybridization, chromosome doubling, recreated synthetic N tobacum. Wheat triticum, how you will be finding over here? Common bread wheat is hexaploid. 2n, though we represent is 2n, but this is 6x basically, 6 times the chromosome numbers you are going to find. So triticum monocum, 2n is equal to 14, you call it as A. This particular species BB 2n 14. Combine this together, this will be, or this one more species over here you will be getting DD. So hybridization, what you will be getting over here? AA, BB, DD. One more hybridization, tetraploid wheat, hexaploid wheat. This is the result of this particular part. Easy to understand, you must be understanding this. Hai na? More you will be understanding that how these genes are getting transferred in the next lecture, laws of heredity. More things will be getting clear over there, and very important conceptual lecture series is going for unit number 1. If unit 1 is solid, unit 2 will automatically become solid. Okay, this is the way you will be understanding. Then there is one more example, again same, you can understand AA, BB, CC. These type of hybrid species can be formed, and these are nothing but alloploids. Okay, alloploids, right? So a difference of autopolyploid and allopolyploid is highlighted over here. Try to understand it. Things which we have discussed is same over here. More things, don't try to get confused right now that what is inheritance, disomic inheritance. These all things will be clear once you will be completing the unit. Okay? Because see over here, you have this particular whole chapter on heritability, sterility, inheritance. We are going to see this, है ना? Right now we have seen the polyploidy, euploidy and aneuploidy part over here. This is done. Chromosome structure is done. Chromosomal aberration in detail. I have told you that how, or I have given you the information about how these aberrations are exactly happening, causing the different types of chromosome structure over here. So main parts are getting over here. Okay, this particular part is over. This particular part are over. Now the next part, which will be combining all these knowledge, will be laws of heredity. That how things are getting transmitted. Why from a particular AA gene, one A gene is coming, from BB gene, one B gene is coming. This you will be getting in the next lecture, that is laws of heredity. Mendel's laws of heredity, which he propounded in 1800 something around, are still valid with a new terminology. This very conceptual lecture in the next class we will be seeing. Then we will be understanding mutation, linkage, heritability, cytoplasmic event. Then these are small topics. Hai na? Major topics are this only. So over here in this particular class, we have understood the chromosomal aberration. One on the basis of structure, second on the basis of numerical. Numerical, if it is getting one chromosome, if it is about one chromosome, aneuploidy. If it is about the whole set, this is euploidy. Under euploidy also you are having, if it is one set of chromosome, that is monoploids. If this is half, then haploids. If this is double, diploid. If this is multiple, polyploid. In polyploid also you have autopolyploids and allopolyploids. What is the difference? You tell me. In structure you have understood based upon the change in gene number, addition, minus, duplication and deletion. If there is a sequence in change, you will be finding inversion and translocation. So this you have understood in this particular lecture. I hope you must have understood this. If you have any problem, if you face any problem, try to contact. I have given you already my number. You try to contact with me. In the next class, we will be moving ahead, guys. Thank you so much. We'll be meeting in the next class.